very interesting. He's simply put, he's going to talk to us today about network analysis and an application of it uh, in the area of epileptic seizures. I'll read this phrase out from his website, I think, which says it very well. His research is focused at the point of convergence where statistical and machine learning theory and methods support human endeavors enabled by computing and engineered systems. And you say that's frequently from a network based perspective, but I guess that means that leaves the door open to other uh, things. Certainly I know that over here there's a lot more interest in network analysis lately. And so uh, I've been contacted by people who want to do network analysis. We had a speaker here last year, uh, but my question was, how come there's no uncertainty estimation? And I noticed that that's something that you yourself have talked about. So I hope that you will tell us something about that today, perhaps even in passing. And besides that, Eric actually has a very illustrious career behind him. He's very modest, so one wouldn't know. He studied at Stanford University, and he has trained over 20 PhD students in his career, which is really remarkable. Written several publications and a number of books as well. In particular, what caught my eye was a book on network analysis um, with R in R, and I think you are teaching an accompanying course in the winter as well. And that book is available for free via McGill libraries. So anybody wants to learn network analysis, that's I think a good place perhaps to begin. And yeah, without further ado.
Okay, thank you, Nandini. All right, so I will talk about networks. I'll talk a bit about uncertainty. This won't touch as much on what we've done with uncertainty quantification for networks, but you'll see that dealing with noise and network data is going to be what is a theme throughout the talk. So I'm going to give a little bit of background, and then we're going to talk about how from fundamental neuroscience measurements do we construct a network. There are literally hundreds of choices these days. We have our own version that we created maybe 15 years ago. So a lot of what was used here does that. And then I'll talk about two directions that we've worked on in terms of taking that as input and trying to address certain problems. One is so-called community detection, and the other one is trying to infer what are called percolation regimes. Okay, so a little background on epilepsy. I'm not an epileptologist, of course. I'm a statistician. I've had the pleasure of working with people in this area for about 15 years. This started with a chance meeting with a postdoc, Mark Kramer. He was a postdoc at Boston University in 2008, 2009. And he said that he was getting into networks. He heard that I had been into it, and he wanted to know, could we do networks in neuroscience? So I guess so. And we've worked 15 years together now. So this is really kind of a story of what we've done over 15 years, right? But it was motivated by the disease of epilepsy. So this is one of the most common neurological syndromes. It apparently affects about 50 million people worldwide. In about a third of these patients, you can't control the seizures, right? The go-to is, of course, medication, but that doesn't always work. So it's increasingly recognized that epileptic seizures are representing kind of an interplay of neural dynamics. They used to be thought of, as I understand it, to be sort of a wave-like phenomena. There were people in dynamic systems that had models that would talk about the seizure being like just a single wave. And then that was starting to break down when Mark and I met. And that was why he wanted to use notions of networks. So you could talk about parts of the brain experience seizure, and that's spreading to others, but then maybe earlier parts leaving that, right? And that's much more of a network-based perspective, right? Please. Be easier on me too. I'm trying not to listen to myself. Okay, so here's our goals. I'll keep talking. We'll know when this goes off, right? So at 10,000 feet, and this ties to a NIH award that recently finished, that was sort of the culmination of this vision that we were working with. There we go. I can feel my own brain kind of relaxing. We wanted to identify principled surgical targets, right? If someone is not reacting to medicine, then the only recourse really is surgery, and it's surgeries that have gotten more refined, but essentially are resective type surgeries that go back about 100 years or so. We'd like to be able to predict which patients will and will not benefit from surgery. And we like to perhaps propose alternative surgical and control strategies, right? And the working premises here is that if you take data that is typically collected in these contexts, in the context of observing a patient, preparing them for surgery and during surgery, right, that you can instead transform that data into networks. And in that representation, that network representation will be useful for trying to answer some of these questions. Right. So here's our work, right? We're using invasive brain voltage recordings, hence why these are for subjects that are already undergoing surgery. People are not going to be volunteering for this. This is patients with intractable epilepsy. We infer dynamic functional networks, are what they're called, during spontaneous seizures. And then we use these networks to inform several interacting streams of research. And so Mark Kramer is the third one there. Sid Cash and Kat Chu are at MGH in Boston. All this is a Boston-based crew, since that's where I was before I came to McGill. The vast majority of them that then have BU/something are former students and postdocs. And you can see that having done this research, that they've gone to sort of a variety of fantastic places. All right, here's a pictogram of what we're thinking about, right? So you have your data on the left-hand side there. I know people on Zoom can't necessarily see the green dot. Moving along, what we do is recreate dynamic networks, right? The data themselves are multivariate time series. They're typically with electrodes that are implanted in a grid on the cortical surface for what we're looking at. We have also looked at data, for example, that's from the scalp. So the methods we're talking about are fairly general. From this data, which are in our case, they're ECOG data, they're very fine time resolution, right? We have an ability to then create dynamic networks using intervals. These intervals then also indexed in time. So we get at a coarser time resolution, a time series of networks from a finer time resolution of multivariate time series, right? We do two different things here. One is to try and find clusters of nodes that somehow are related. They're more clumped together than others. It's the analog of doing clustering in data. This is in networks called community detection, right? The other thing is to then try and describe those communities. Are those communities all dense? Are some of them less dense, more dense? Does it change as the seizure begins onset, as you continue through the seizure, as you finish the seizure, right? And does that in any way help inform surgical targets or possible clinical outcomes, right? We also are very interested particularly in what is called seizure onset or similar types of things. So it was understood that when the seizure first onsets, you typically get a much greater degree of connectivity in the network, and I'll show you some of that. When you have a plot like I have on the bottom there in blue, of simply the number of nodes that are in the largest connected piece, right? What you'll typically see is that there'll be a sharp increase. This is called percolation in the network literature. So that's a percolation curve. The fact that those percolation curves seem to correspond to onset of the seizure and some of the internal seizure dynamics led us to want to focus particularly on that. And there's lots of models for percolation. So it suggested that maybe it would be interesting to see if there were some mechanisms that were more associated with seizure onset than others. That might then in turn suggest possibilities of what might be going on in terms of dynamics of the seizure. All right, so let's talk about how we get our networks, right? And then I'll talk about these other two pieces that we're forking in that diagram. So our data are ECOG data. These are assumed to inform multivariate time series. They are at roughly millisecond resolution. And for the subjects that we have, these are usually being observed for at least 24 hours. Okay, so it's very long data. It's about 80,000 time points. Okay. From these, we construct a time series of networks. These are usually indexed in our work at a resolution of 1 to 5 seconds. This is based on a great deal of hands-on work, looking at moving windows, overlap of windows, trying to get some sense of independence between the windows and yet still capture the text. That is sort of a subject of its own talk and discussion. Okay, so I am going to be going just high level through most of these things, and I am going to be going somewhat quickly in the sense that most of these are sub areas themselves that have whole subliteratures associated with them. Okay. This was an early paper. I think, in fact, it might have been the first paper with Mark and company in 2009. Here we get with confidence. That's beginning to get at the topic that Nandini mentioned of uncertainty. We had a basic goal here. Right. This basic goal was, if I'm going to define a window, take a bunch of time series, somehow define a notion of association between nodes, which for us were going to be corresponding to locations on the brain, and then we're going to create a network. Right. If we then move to the next 10 seconds and I create another network, how do I know that somehow they are created in comparable fashion? And how do I know in particular that there's somehow not a propensity towards more edges just by chance, for example, in the first interval than the second? Right. And so ultimately that it can be looked at from a testing point of view, and it can boil down to controlling certain types of error. Right. And so we were one of the first papers to try and formulate it this way in the time series setting and provide a general method that would allow us to create slices of a network time series that all would be controlled at similar error rates. And therefore, you could begin then to start treating them as sort of a continuum of networks that were comparable and ask questions across them rather than needing to stay within slices. Okay. All right. So what do you mean by coupling in a network is fundamental to doing any kind of network analysis, right? Networks are meant to represent relations. Right, relations can go from Facebook friends to network connectivity to lines between stations in the metro. I mean, it's an enormously broad area. When you are having your network be constructed or inferred from data, you have to establish some notion of what you mean by coupling or association or something like that. There are many, many versions in this area along with time series, right? A standard one is to say that by association, we mean something like Pearson correlation, but these are time series. So the analog would be something like maximal cross correlation. All that's doing is taking the correlation of the time series with its lag, and then you look for the largest value of that as a function of lag over some number of lags. Okay, so that's maximal cross correlation. So we've chosen that to be, in most of our work, our notion of coupling. And then what we're doing is, for every pair of electrodes that are on the surface of the brain, we're asking the question in a given window, is there no coupling, or is there evidence suggesting coupling during that time period? Right? And so no coupling becomes our null hypothesis. And testing when is that cross-correlation statistic, quote unquote, large, is the essence of trying to figure out when I assign an edge to two regions of the brain. Right? And so what that means then is that you're taking the signal from one electrode, you're taking it from another in the same window, and then you're looking at their lags. And if you can get them to line up a bit in a reasonably strong sense at some lag over a reasonable lag, then you say, yeah, there's some sort of correlation between them. And that's a very standard time series notion. We're just adopting it here, right? When you do this, you have to do a few things. You need an appropriate test statistic. You have to be able to accurately compute p-values under your null hypothesis, right? You're being frequentist, for example. And at the same time, we're going to do this not just for two nodes, we're going to do it for all nodes. So you have many, many tests, so you have to be able to control for multiple testing. Right? And that's the essence of the paper that I showed. Right, this is a quick dive into notation just to give you a sense of what we do. So first of all, we take so-called Fisher transformations of the cross correlation. Right, this ends up essentially making them more Gaussian. Right, then we're looking at the maximum. Right, we take that maximum, we standardize it by its variance, so it's normalized. And then we want to be able to do a p-value type calculation, but there's a maximum in that. Prior to this paper, something that folks did in this area is they compared that statistic to a Gaussian because we compare everything to Gaussians, right? But when you look at maxima, those are absolutely not Gaussian, right? The simplest way to look at it is to take a bunch of Gaussians on your computer and then take only the maxima and look at their distribution over many trials. You see that they're all lying out in the tails, right? And so these are so-called extreme value distributions, and you can get an approximation to how a p-value should be calculated using formulas like this from the so-called large deviations literature. And the main thing to note is just that tail probabilities look like e to the minus, right? That's symptomatic of a tail, and that's symptomatic of what you call large deviation or distributions. Okay. This is one of my few proofs and pictures that I'll have throughout the talk, right? Again, we're not going to do a deep dive into formulas or whatnot. I just want to tell you a story that ranges 15 years, right? And give you a sense of what we can do. And as I said, most of this stuff can be done in other contexts. And even here, we have a lot more we'd love to do, but really moving much more towards the outcomes direction rather than the basic development direction. We're sort of done with the basic development. We're really at a point now where we could really move this into large cohorts, probably retrospectively, presumably, and do some interesting analyses. So this is a network. It's pretend. The data that we generated as time series were generated from a biophysical-based neuroscience model. So it has some complex dynamics to it. It has some complex noise to it. And they, being somewhat engineering, like to look at power spectra. This is basically to show you this is not just Gaussian noise. There's signal there. What you want to do is from the time series we generate, recover these edges. If you use the method I talk about and compare it to your usual normal distribution, you get a network that has a grand total of 0 edges. It's because you're completely using the wrong thing to calibrate your uncertainty. If instead you use the calculations I showed, you get something like this. This is a false discovery rate plot where you accept everything that's underneath the line and you reject everything above the line. So this is corresponding to edges, and those are where the edges are. Right? There's also a way to use bootstrapping the fundamental data. It's much more computationally intensive, but it's certainly a sound way to go. And you see you get essentially the same difference. So with a little bit of math, you can save yourself time and get quite accurate. Okay, so this is what we use throughout the rest of the talk, right? Or variations thereof, right? We apply that, for example, to 24-hour networks over multiple subjects, leading to network time series on the order of a few hundred to a few thousand for a given day. Okay. And then we want to understand those as data objects. So the rest of the talk, we're no longer worrying for the most part about the original time series. We're just thinking about network time series and wondering how do those evolve. There are groups of edges that suddenly spread further downstream in time and get much more groups, or do they disappear and become not groups? So think about if you were spatial, temporal spatial, you're thinking like tubes that are coming together, tubes that are growing, tubes that are shrinking if you were looking in real time. Okay. All right, so what motivated that? A 2010 paper motivated that idea of tubes. We took what we had done in 2009 and we applied it on one subject to 3 seizures. Here is what it looks like if you do little snapshots, right? So the red is meant to be a bunch of nodes that are all connected. The yellow is the next largest set of nodes that are connected, but they're not connected to any red nodes, right? So these are called components. So the red is the largest component, the yellow is the second largest component. It's a very crude way of thinking about clustering, but it is a way, all right? If you take a look in the time course of a seizure, which is on the bottom of the upper left-hand plot, right? You see there's some gray bars going up on the left and going up on the right. That is the onset of the seizure and the termination of the seizure. And what you see here is that we've computed 1, 2, 3, 4, 5, 6, 7, 8, about 12 time points we've computed networks, okay? And we've looked at how many components are there. And so you start off just before seizure onset, which is determined by a clinician, at about 6 components. It dips slightly, and this is on average over a few different seizures. And then it goes up during the seizure, right? So you're at around 7 on average. And then it dips down very sharply towards the end, right? So this is where the title of the paper and focus here of coalescence and fragmentation. The idea that during the seizure, and I mentioned earlier, hypersynchronization was what people thought was a good model for what was going on in a seizure, right? Everything synchronized in terms of the brain signals. And it was like one big wave passing through and just collecting everybody. But these data suggest that that's really not what's going on. Instead, there's coalescence where where nodes are being recruited, and then there's fragmentation where they're peeling away, right? And this is giving you a very rough sense of how that looks. If you look over the first row of networks, that's early. Middle, you see that there's a much smaller red zone, for example, and later you see that almost all the nodes are now recruited to red. Okay, so the whole point of the paper is it's more subtle, right? It would be good to be able to try and explore that. So we applied to the NIH a few times. And eventually they agreed that this would be a good idea to explore it, and we had possibly some good ideas to do. And so that's what these last 2 projects I'll tell you about are. One is the method of so-called dynamic flux percolation. And then the second is this methodology for percolation. Right? So if you are dealing with time series of networks and you want to do something a little more sophisticated, then carve out subsets of nodes that are connected from all those that aren't. Right? You'd like to allow, like, what happens if I have a group of 5 nodes and a group of 10 nodes? They're each all connected within themselves and never between. And then all of a sudden, how would it happen? Maybe by noise. You have one node in each of those that are connected. Right? What's the difference between 2 big groups and 2 big groups joined by one edge? Right? For most things, not a lot. From the point of view of clustering of data, right? The analog here would say not much. They would call those communities instead of groups. And they would say that really you should still declare them as 2 using most typical measures. Okay. So this is not this idea of using components is really not a particularly state-of-the-art way of talking about subsets of nodes. When you open the door on that question, it turns out there's lots and lots of methods for detecting subsets of nodes across time, although it is much harder than trying to detect subsets of nodes in a given time slice. Okay, if you start thinking about it, some of the the low-hanging fruit ways of doing it is to do community detection in one slice and do it in another and do it in another, right? And then you just have a bunch. Now you have to figure out, how do I match the different communities you find? Is it the biggest goes to the biggest, next biggest, right? So it becomes non-trivial as even if you know how to do detection within slices, right? So we have a methodology that defines a community explicitly as subsets of nodes within it and across time that are reachable by walking little teeny shapes. Right? These little teeny shapes are called plexes, and they are chosen particularly to be robust to noise. So again, going to Nandini's point, we're not quantifying the uncertainty, but everything we're doing is trying to be able to help take into account. So this is generalizing an earlier technology in this area in 2 ways. That methodology would take, for example, triangles, or more generally what they call cliques. And if within, say, one graph, I could take that triangle and flip it on one of its edges, and I could find another node where there were 2 edges going to it, and I could flip it again and find another one where every time I was always flipping to a triangle, then if you carve out the extent that you could walk that triangle, that would be called a community, for example. That's very brittle. The reason being that if you're forming networks under noise from time series, you might, by type 1 error, simply not have an edge, right? If you don't have an edge and your triangle should have walked, but there's no edge to land on, then suddenly you get it wrong, right? Fluxes, without going into much detail, fluxes are a generalization that says instead of a shape having exactly this number of edges, it says that every node has to have at least a number. And the at least is just enough to give you some robustness in ways that we spell out in the paper, and we sort of show how it correlates to different forms. Right? This is a way to think about it as pictures. Right? So on the left there in figure A, I have 2 time points t and t plus 1. I have 3 nodes A, B, and C connected by 2 blue edges. And what happens is that between one time point and another, I have that AC edge suddenly become an AB edge, right? And so I first had ACB, and then I had ABC, okay? So the question is, can I walk that shape across time? And yes, I can walk that blue V across time. That's what the little red bars are. The red bars are not meant to be real edges. They're just a mathematical construct to allow things to potentially go across time if there's somewhere to land. So you can think about taking a big step, right? And it's a leap of faith. You're hoping that you have somewhere on the next time point. And then within there, you carve out a community by walking around your time slice. And from every place within the time slice, you can also walk across. Right? So it sounds complicated. There's an algorithmic component that is used to do this efficiently. You don't do it intelligently, it's not efficient. Right? But you can do this. Right? Here's what happens again as an illustration, where in part B, I have a orange square that is a community, and I also have a purple set of nodes that is another community. And all those colors are just edges amongst them. So they've been colored to indicate the connectivity of those communities. At time t plus 1, enough of the edges have disappeared from that purple community that we no longer declare it's just dissipated. On the other hand, the orange one appears to be growing because it's recruited more nodes according to the that I expected. And then, interestingly, it fragments. Right? And now it's fragmented into 2, but we're coloring those 2 groups the same because they came from that original orange. So if you picture a tube that breaks into fingers, right? That's what we're looking for. This coalescence fragmentation. We want to actually track it in time. Okay. Yeah. Please. Yes, all of these are in a single individual, and we can do this on multiple. Yeah. That's right. At millisecond time resolution for epoch data, you get roughly in 24 hours about 80,000 time points in your original time series. When we aggregate up, because we need to have some width of a window to talk about, is there a signal and is it correlating between nodes? Then we get to anywhere from a half second to 5 seconds, depending on what resolution we choose. And then that. So the ABC are nodes corresponding to, trying to think what the word is, not terminals. My mind is blanking. The measurement points on the brain, right? So when you put the measurement grid on the brain there, they're measuring local field potentials, right? So these little terminals are corresponding to nodes. Right. And so the measurements in ECOG, for example, are literally measuring local field potentials in the brain.
So essentially, A, B, and C are being measured 80,000 times for each individual.
They are local field potential for each individual for each terminal.
Okay.
Right. Those are being measured at 80,000 times.
And so what you're illustrating here is how the model that you've built connects these measurements between subsequent time points on the same individual.
So you could think of t as like 1 second, t plus 1 as 5 seconds, t plus 2 as 10 seconds, for example.
Okay.
And those have used that original 80,000 time series to then say in a 5 second window, to what degree is there correlation? Right? And so that would say that A and C have non-trivial correlation, B and C have non-trivial. When it passes to time t plus 1, right? A and C no longer have that correlation, but A, B do, and
Okay.
and that's all done in a testing sense, all controlling type 1 error.
Okay.
False discovery rate.
All right. Okay. So the DPPM is an approach that is robust and able to track real patterns that happen.
That's right. So now that you have those networks, then DPPM is looking to trying to track the patterns within those networks. And it's a very particular type of pattern. I'll show you how it works and compare it to some state of the art. They weren't designed to do this type of pattern, right? We, because of that earlier work, we're looking at coalescing and fragmenting. We very much wanted to be able to have something that could follow splits, right? And that needs the particular type of tracking that we're doing. Right, so here's a simulation. The DPPM is here in the second column. The first column is a way of representing truth. And the third and fourth are what happens when two competitors that are very good competitors, just not designed for this, end up trying to make an attempt at this problem. Right, so what we have here is for a 64 node network, this is just pretend, might regenerate the same type of data that I've been describing. And I've just lined the nodes up vertically here, 1 through 64. So I lose all representation to the brain, right? It's just 64 nodes. And what I say over time happens is 3 different things. In the first one, you start with most of the nodes that are part of it, and then gradually you start losing subsets of nodes until around time 120, you only have about a fifth of the nodes that are part of it. Okay, so that's a large community that's gradually diminishing. And then we have 2 other things going on where, first of all, we have 1 tube splitting into 2. And in the bottom, we have 2 tubes merging into 1. Okay, so that's how you should interpret that. And then we generate the data. From that data, we form networks. The networks go into either of these 3 different methods. Someone has to move. Okay. And then what you see here is you want to recover the look of these light blue squares. And you can see the only method that's even coming close to that, although it has a little bit of background scatter of noise, is our method. Right? And it does actually quite a good job. Right? And if you don't like the background scatter, there's some easy image processing techniques you can do to get rid of that. But those techniques will not get rid of the fact that you have lots and lots of different colors under the other 2 methods. And that's just basically showing that they are thinking communities as something much, much smaller. They're working way too hard to try and capture the same shape. You can see the outline of the form, particularly under CPM, right? It's just not doing a good job of identifying that it's all one thing. Again, they're not designed to have this sort of a sense of following through a tunnel that we have, okay? This is what it looks like if you put this all together for a single seizure, right? So on the top here, this is a subset of nodes shown. These are the actual time series Nandini was asking about. This big line about 2/3 across at where time is marked as 0 on the bottom, that is what was declared seizure onset by the clinician. So, right, so you see a real marked difference in the visual nature of these time series to the right of that line versus the left. We then have a color coding of, again, the nodes are stacked from 0 to 60 again, and we have a color coding of the different communities. And you see, again, the community behavior is very different pre and post seizure. What's going on with all these colors is it's saying that it's just finding little bits of communities, that prior to the seizure, right, there isn't this large sort of pulling together of the whole brain. You have bits of the brain working together, and then other bits, and then other bits, and then other bits, right? On the other hand, once the seizure comes, remember that wave-like analogy, right? You've seen a full-scale recruitment, except for during the middle part where there is some fragmentation, and amongst the fragmentation, there is for a little bit another community color that's recruited, right? And on the bottom here, this is tracking just the largest community, saying how many out of all of the nodes we have are actually in this large community. And you see that it has this stark increase right after the seizure onset. It goes through various types of dynamics, and around 0.40, it again blasts up, right? This is percolation. right? And that's going to be the topic we're going to talk about for the very last part of this talk, right? This sort of steep ramp up, that's called percolation. And we're particularly interested in those because across seizures, we routinely were seeing it just after or just before seizure onset. And we were sometimes seeing what we thought might be a sort of secondary percolation later into the seizure. We wanted to understand it. Okay. Okay, some interpretation here. Done all of this work, you have a community question you can only interrogate. So this is looking over about 30 different seizures, right? In those seizures, we have pre and post. That's in blue and red, respectively. And on the left-hand side here, what we see is the maximal community tends to be larger and of longer duration during the seizure than before. On the right-hand side here, we have angle 1, 2, angle 3, 4. These are associated with outcomes. Angle 3, 4 indicates worse surgical outcomes than angle 2, 1, 2. And so this is saying that worse surgical outcomes exhibit more communities with longer maximal duration during seizure. Right? So this is showing that after all that work and using the communities now as high-level descriptors of what's going on in the seizure, you can those communities, and they are correlating with both pre and post seizure, and they're correlating with better and worse surgery. It's a small data set, but it's suggestive that it's something that one can do on a much larger data set, either in this setting or on other sorts of settings now that you have all the technology there. Okay, last bit here. So statistical inference of percolation regions, right? We're particularly interested in this phenomenon. This is an old picture. I think it's one of the first ones that Mark showed me around 2008, 2009, right? This is, again, number of nodes in the largest component. So we're back to fully connected, right? And it's showing, again, a very sort of random walk-like flavor until about the dotted line, the vertical dotted line being seizure onset, where you get this swoop up. Okay, so we notice this time and time again with epilepsy. And if you're in the graph literature, the random graph literature, whatever you look at that, that's a phenomenon that's been studied in detail for like 50 plus years. It's called percolation. In the early 2000s, there was a whole explosion of additional work just in that area where people defined dozens of models to modulate just how steep that climb was. And those models often corresponded to mechanisms like avoiding having big components get together until the last second would give you a much steeper climb, right? Facilitating them earlier would get a much slower climb, right? And additional sort of models, right? So we're at a time point where we had a bunch of mechanisms. They were simple, right? They're not biophysical, but still they could be potentially useful. And we had data, and what we needed was a statistical method for us to try and be able to test models and say, do these data look more like one mechanism could produce or another? All right, now here comes uncertainty again, right? I did not work with Nandini to coach this, but here we go again, right? So this was a paper of sort of an intermediate result in the mid-2010s. What we did is we looked at what happens if you take two different types of percolation. One is for a random graph model in the classical sense. All you do is randomly choose a pair of nodes who do not possess an edge, and you assign it uniformly, okay? That has a certain curve. That's the blue line, okay? On the other hand, if you hold off as long as you can, try and keep matching small and small and small, and finally you get those big ones and your big component snaps into a massive size. That's what you get with the red line. You see how much steeper that is. Okay, so it's just two extremes in the mathematical literature as to how to do it, and you can see how distinct these curves are. On the other hand, if we add noise, where some of the edges aren't real edges and some of the non-edges are not real non-edges, then you get the blue and the green. In other words, you can't tell the difference, right? At least not using curves, okay? So again, high level, this is work that just appeared earlier this year, a few months ago. It was long in the making, 2 PhD students working on it in successive phases of their theses. But we noted that for a number of the random graphs, they form Markov models. And if you have the idea that the network is evolving and every once in a while on a regular basis, you measure it, just like our time series every 5 seconds, then what you get is a sampled hidden Markov model of graphs. Okay, so I'm not going to go into a lot of the details, but this basically says that every time there's going to be a change, do you add an edge or delete an edge? Right, and then that's going to produce a different network. And then every so often you observe a network, but you observe it where every single edge is observed with some noise, meaning a coin flip that you don't see it. And every single non-edge is observed with some noise, meaning a different coin flip that you don't see. Okay, and that's our model here. Given this observed graph, what we want to do is if we have one of these for one possible mechanism and another for another mechanism, we'd like to compare them. Okay. So what do you need to do that? You need to develop an EM algorithm for parameter estimation. Each of these models have some parameters. We use a Bayesian approach, so you have some weak priors, and we use Bayes factors to assess the evidence between the so-called classic one, which is Erdős-Renyi, and this other one that holds off, which is a so-called Aklioptis model. Right? The EM algorithm is decidedly not trivial. This is why we work through 2 theses to do this. It uses data augmentation, particle filtering, and a number of computational speedups because when you're trying to explore graph space, you're in a very, very large space. Okay. That's what the algorithm looks like. It's a paper if one wants to really delve through it, but there's basically things like EM iteration going on here. There is sampling of particles, sampling of paths. The reason it's hard is because you never observe this. And so what you have to do is you have to basically, from these observed time points, you have to infer possible paths between these. This is not so hard to denoise, but it's the fact that you don't observe the times between that makes it a very hard problem. Okay, so finally, in wrapping up here, if you put it all together, what we did is you've seen something like these first 3 plots. Right. What we did then is we created an automatic percolation segment finder. It's not that hard. It's basically based on slopes that are high enough in a sustained period of time. And you see some dark blue just after 50, some dark blue between 200 and 250, and some dark blue after 300. Those are the 3 areas that our particular segment finder decided to say, those are pretty free and clear areas of percolation. It'd just be nice to figure out which type of model best describes it. It's not saying there aren't other areas that go up, right? The thing's constantly going up and down, but you're trying to distinguish between what might just be random walk dynamics or other type versus stuff that clearly was down and then took a random. Okay. So we did this, right? Taking input as our ECOG data from a human subject on 3 seizures. In those 3 seizures, they had a handful of segments in each of them, so not many. This is very small data just on a single subject. And we concluded that this classic Erdős-Renyi percolation was better supported for the entire seizure 1, the entire seizure 2, and the early stages of seizure 3. But this other model was better supported for the late part of stage 3. And so it doesn't permit you to infer a lot based on one patient, right? This is meant to be a proof of concept. But you could say clinically seizure 3, it turns out differs from the other 2 seizures, right? So I hid this, just the magician part, right? Seizure 3 remains focal. So focal seizure versus a generalized seizure, right? They start locally. If they stay locally, all of these start in the left hemisphere of the brain. Right? If they stay that way, you call it a focal seizure. If it spreads across the brain, then it's generalized. So it turns out that the only one out of the 3 that was spatially focal is also the only one that exhibited this differentiation in the early and the late. So it suggests a hypothesis, remains to be tested, right? That compared to classical percolation, the local PR percolation could correlate with focal seizure termination and prevent generalization, perhaps. This is what I mean by saying that, this is 15 years of work with an idea over coffee of wouldn't it be cool if we could do all this stuff, right? It's taken about 15 years worth of development just to even say, what do you mean by network? And oh, we have to get the noise under control. We're going to be able to talk about how it changes across time. Then being able to talk about what we mean by communities in time, and then how they ramp up. So we have several tools now available. It's a set of tools that are all available online, GitHub, and I'd be happy to talk to anyone who might want to talk about what we might mean to say, move this into a retrospective type study or something like that. Okay, and I'll stop here for questions.
All right. Yes. All right. Ian, you've done some time series work, multivariate time series. Yeah. Yeah. Indeed, I am just indeed trying to understand. So, I mean, this is an application in what's it called, in epilepsy particularly, but I presume that this is generalized to other areas.
They could certainly generalize. Yeah, certainly generalize. I mean, I've worked a bit in fMRI data, G, and one of the things we know with any of these sort of data is they're always trading off temporal and spatial. So necessary. Right? And so depending on what data you're working with, some of these questions may be more or less relevant. So for example, we did a study in MEG data where it was task-based work, and they were very interested in really understanding exact time points and delays and things like that for something to happen. The something was at very necessarily coarse spatial notions of brain regions. With 3 to 5 to 10 data points, you're not really going to get very interesting tubes in terms of dynamic community detection. But for that question, that was the right data. Right. So we've been very careful based on just a handful of seizures not to try and make any high-level recommendations, right? The closest that we have so far is this, right? So that was based on about 30 subjects, right? 30 seizures, I believe. I don't remember how many subjects. Some of them are multiple. That's beginning to show some connections here between outcomes, for example, and characteristics of the networks, right? The best way to think about what we're doing here is if you believe in the sort of trend in neuroscience over the last 20 years to take the notions of networks and network connectivity as depending on the modality you use and the way you construct it as being perhaps usefully representative brain connectivity in terms of, say, communication patterns or something like that, then going from basic measurements to networks is a useful step because at that point you can treat the network data objects as your fundamental objects that you want to now analyze. And you ask questions of those objects, which tend to be different questions than you would ask of a time series or a spatial data set or something like that, and see if that can correlate with questions of interest, say in terms of outcomes or what. I always tell students when I teach, if it's not a network question, don't use it. Right? There's questions in neuroscience, for example, easily done by covariance or tensor-based methods, for example. You don't have to actually play. But there are other things that have to do with clumping and paths and things, and those are just network concepts. So depending on what your questions are, these types of things could be relevant. And in principle, yeah, they can be used. You can put any data type through. It's just a question of time and space resolution may make some of these analyses more interesting or less interesting.
Quick question. Sorry. Thank you for your presentation. I just wonder, is this for exact prescription at that time, or it also can predict, since you have three seizures and the patterns, you can predict in future how the seizure looks like and network.
Oh,
And also, yeah.
yeah, it would be interesting to try and do prediction. I think you would need to repeat some of what we've done on a much larger cohort first and bring in covariates, right? And that's something we haven't done. So we haven't done any, and this is sort of like a far end of the goal of the NIH grant, but that's grant is done. But that was the idea that if you could bring in, say, dozens of seizures and at the same time have the various characteristics of the individuals, social determinants of health, various things like that, that alone could be an interesting study, right? Because what you can do when you have these network time series is you can think of any statistic that you'd like to create as a function of this time series, right? You pull off, the largest time something, first time something happens, whether one time does or doesn't predict 10 seconds forward. You can get any of these as you want if they're network-based questions. And then given various other characteristics of individuals, you should be able to put that into standard statistical modeling, say longitudinal type models, GE type of things and whatnot. And then
Similar ideas that you can have the Cox pH time series versus combined with the time joined modeling with the longitudinal the modeling, you can get the hazard ratio across the time.
Sure. So we could take some simple examples. Say you do all this stuff with communities, and then you count how many communities are there at any given time. Now, all you have for every seizure is a time series of counts, for example, right? If you ask more complex questions like how many times, within how many times you have one community that eventually breaks into two, and what's the wait time for that? That might be something that looked more like a hazard type model. You have to think about it, but I'm just throwing ideas at, but yeah, that's any of that stuff, and that has a long history in neuroscience, not of those particular types of things, but like with panels of neuro data. To say that I take, say, I do fMRI at 3, 6, 9 months, for example. So I just have very simple measurements, not really time series. You take your favorite descriptor of the network, and then you throw that as the response into a longitudinal model for a bunch of subjects. So they do this like on the ADNI database. It's like
Program. I presume you've done this program. Is that right?
Oh boy. Much of it's in MATLAB because Mark's group was in MATLAB. The last project I believe is in R. I think the middle project with the tubes, I think that is in MATLAB with C written underneath. Because of the computational complexity.
I think one thing that Ian and me know is that whenever you have those latent variables, it's going to tremendously increase your computational time.
Exactly. So particularly for the middle one, the middle project, the one with the DPPM. Yeah. So that's like a whole separate conversation. There's certain parts of that problem that are NP complete, meaning that you have no efficient algorithm to do it. And so then you enter the computer science world of approximate graph algorithms. And so there is a class for these, and we have adopted one of those and modified it for our purposes. And then even that you want to have be efficient, and that's why there's C underlying MATLAB doing a number of these. Yeah.
Like how long does it take to analyze 30 seizures?
That's a good question. I think it takes at most on the order of a few days.
Yeah. All right. That's it.
But again, keep in mind, this is 80,000 data points per seizure. And once you have those networks, you have the networks. You're done. Right. So you do it once. Yeah. Okay. Thank you all.